This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 199, recorded on September 13th, 2012. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today from North Central Florida, Rich Condit.
Howdy, Vincent. How are you today?
I'm just fine. Beautiful day here. Really nice. 26 degrees and sunny here in New York City.
No kidding. You probably got a better day than we do.
What do you
We
got rain?
27.7 Celsius. And mostly cloudy.
No, that's the way it goes.
That's okay.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How's your weather?
It's gorgeous. It's 81 Fahrenheit, 27 Celsius. Just very, very pleasant. A few puffy clouds I think I saw earlier, but mostly clear.
September is a good month for weather.
Usually is, yeah,
Here
in this part of the country.
Here in this part of the world. I think down south it probably isn't so good.
It's starting to cool off here. I can see the light at the end of the summer tunnel. So that's good.
Summer's over next week, right? A week from tomorrow.
Is that right?
21st.
Yep.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
How's the weather out there?
Pretty much like what Alan described. 81 and 21 C, or as my younger brother likes to think of degrees C as degrees Canadian. So it's
degrees.
Yeah.
Where did that come from? Just a big C?
I guess. We're pretty close to Canada. And
they use it there,
Yeah.
Someday we'll get to your brother's letter.
that's my older brother, yeah.
Oh, you have two brothers?
Yes, yeah.
Are you in the middle?
Yes, I am.
Wow. Where were you from? Where were you born?
I was born in Illinois, but grew up in Ohio.
I don't think we ever got that out of you, did we?
Probably not.
Like your first episode, we didn't ask you where you were from. Born in Illinois.
So like totally Midwest, right?
Totally.
Yeah. 26 years out of the Midwest, and here I am again.
And you're back. I was with your colleague, David Aronoff, the past couple of days.
Oh, cool.
Out in San Francisco. He's a good guy. We're going to get him on one of my other podcasts one of these days.
He'll be good.
Talk about C. diff. We chaired a session yesterday morning.
Mm-hmm.
It was fun. All right, today we have a big agenda. Probably be 2 hours, I would guess. All right, and it's good because our next episode, number 200,
Woohoo!
we are somebody left. Someone left.
Not me.
Not me.
You can guess who.
Alan dropped off of the internet.
Is he going to do this for the whole episode?
I hope not.
First edit.
Sorry.
Did you sit on a switch or something?
Yeah, the digital equivalent. I was going to take a drink of water, so I hit the mute button, but I accidentally hit the hang up button instead of mute.
I've said, his modem is gone again. But
No.
that happens all the time. I hear podcasts where I meant to hit mute, but it's silent.
They're right next to each other. It's
a problem. I was saying, I don't even remember. What was I saying? Kathy, you'll know.
We have a big show.
We have a big show.
Yeah,
Sorry
big
about,
show.
yes.
Next week. So we're doing a special episode for 200, and we're going somewhere, and there's going to be video. So it may take a little bit longer to release. It may not come out on the following Sunday. So we make a big episode today, and you can split it up if you want.
Great.
Right? Is that a good plan?
Sounds
Great
good to me.
Mm-hmm.
All right. Some follow-up. We have an email from Nels Eld. A note of gratitude to you and your crew for generously interrogating my recent paper on the experimental evolution of vaccinia And Nels, of course, is the senior author, the PI on the poxvirus accordion genome paper that we did I guess it was 2 weeks ago.
Mm-hmm.
198. By the way, it was evolutionary biologist Lee Van Valen, not Richard Dawkins, who proposed and coined the Red Queen hypothesis in 1973. Lee was a true character and early advocate of open access publishing. His original manuscript on the Red Queen was published in Evolutionary Theory, a journal he created and self-published at the University of Chicago, still available free and now online at leevanvalen.com, along with some interesting stories about this unconventional and brilliant scientist who recently passed away. Count me as a new listener and fan of TWiV.
All right.
Cool.
I didn't say it was Dawkins.
I think I did.
All right. No problem. It doesn't matter. I just wanted to note that. I think I had said who did it, and you said, yeah. Okay.
Yeah, I associated the idea with Dawkins because I think he's talked about it, but I just credited it to him without checking my facts.
We had 4 emails.
Yes, we have subsequent emails on this. A lot of people noticed this error.
It's good. Means they're listening, right?
Yes.
So, Rich, did you want to have an afterthought on TWiV 198?
Yeah, I had 2 afterthoughts. This is a little high end, but what the heck?
It's not for me. I'm going to go get a drink of water, okay?
Just don't hang up.
So I don't want to go through the whole experiment again. If this doesn't make any sense, you just got to go back and listen to the previous episode. But you'll recall that there were 2 vaccinia antiviral factors, one E3 and one K3. E3 binds double-stranded RNA and knocks it out, and K3 is a homolog for the target of this kinase PKR. And they knocked out E3, and the virus has a host range, and the virus overcame that evolutionarily by making a whole bunch of the K3 to knock out the PKR. That implies, okay, so E3 binds double-stranded RNA. Double-stranded RNA is a sort of a global danger signal for cells. Can activate other pathways like the 2,5-A pathway and stuff like that. So this virus is missing that, and it can overcome the restriction just by overproducing a protein that swamps out PKR. That implies to me that PKR is the dominant pathway in these cells, as opposed to the other double-stranded RNA pathways. Now, that's just in these cells. But that interests me, and it brings up the other question, what if you do the reverse experiment and knock out K3 instead of E3 and ask whether the virus can overcome that problem? Does it do something with E3? Does it do something with something else? Is it out of luck? I don't know. So, Nels, get on it.
So, see, both K3 and E3 bind double-stranded RNA?
No, K3 does not bind double-stranded RNA. Right. K3 is just a phony substrate for the PKR.
I remember from the paper that they said K3 is not very good at antagonizing HeLa, PKR, whatever it does. So that's why they left it in there, I assumed.
Right, well.
And so if you took it out, E3 would be really good, so the virus probably wouldn't grow well, or it would...
I looked this up. If you take out K3 and leave only E3, you can get a host range. In fact, it's the complementary host range of the K3 mutant. At any rate, you can get a host range phenotype. So the K3 mutant will not grow on some cells, and it would be interesting to see now whether the virus can compensate for that.
Yeah, maybe E3 will expand, right?
Could be,
or something else. Yeah, it's a good experiment.
Yeah.
Okay. All right, that's your follow-up. We had a note from Marion, who also... Marion sent us an entry from Wikipedia explaining that the Red Queens came from Lee Van Valen, and we had actually cited this in our show notes. Tommy wrote to your TWiV crew listening to 198, and the Red Queen hypothesis of antagonistic coevolution was brought up. I thought I should make a correction regarding the originator of the Red Queen hypothesis, and he gives us links to Lee Van Valen also.
Yes.
Love the show. Keep up the good work. P.S. I listened to the end, so I know that I shouldn't be hanging out for a show this week. Excellent. Tommy's one of the people who listens to the end. There are actually a couple of them.
Good.
Next one's from Adam. Howdy TWiVites. He also
Corrects us
on the
Red Queen
Dawkins
hypothesis.
Red Queen. It mentions a book called The Red Queen, Sex and the Evolution of Human Nature by Matt Ridley. And in fact, I think that was a pick some time ago. Let me pick it up where he says, I have to thank you for making this mistake, as it has led me to actually read Van Valen's paper in full, which I had not done before, despite having cited it in a number of undergrad essays. There is something really enjoyable about reading old papers where the authors wax philosophical on their topic, and it is a nice change from the dense, ultra-concise papers that are standard these days. Whether this was because they had less data to cram into figures, fewer authors, or the journals were less pressed for space back then, I do not know. Maybe all of the above in this case, seeing as this article was the first edition of a journal that Van Valen founded. Probably because I have more pressing uni work I should be doing. I spent quite a bit of time reading about Professor Van Valen, who died in 2010. He was a pretty amazing guy who wrote on many subjects, taught and was beloved by his students, contributed several original ideas to his fields, founded journals, and even wrote explicit songs about dinosaurs. Nice. All right, he makes him a pick of the week, which we'll mention later.
Yeah, I read some of this stuff. It's the guy was crazy.
Adam is
in a fun way.
Crazy and crazy smart, yeah.
Yeah.
Adam is from Melbourne, Australia, 18 degrees Celsius with a chance of rain. P.S. If nothing else, go check out the acknowledgement section of Van Valen's A New Evolutionary Law.
And I looked that up,
Yep.
where he starts out by saying, I thank the National Science Foundation for regularly rejecting my honest grant applications for work on real organisms, thus forcing me into theoretical work.
What's the reference to St. George?
That far I did not go.
He must have said the same thing.
Could be.
Okay, the next one is not to do with our mistake.
Yes.
It's from Josh, who writes, Dear TWiV Doctors, I have a question about the paper discussed last week on the emergence of fatal avian influenza in New England harbor seals. What I cannot figure out is if they were looking for an influenza virus from the start. I mean, I assume if you sample a bunch, 5 in this case, of seals, you're going to get a bunch of viruses. Did they report the detection of any other viruses in the seals? I listened to your segment again and read the paper on MBio. The paper says this: nucleic acids extracted from lung, trachea, liver, kidney, thoracic lymph node, mesenteric lymph node, spleen, skin lesion, and oral mucosa were tested by PCR for the presence of a wide range of pathogens, including herpes viruses, pox viruses, adenoviruses, polyomas, callices, paramyxos, astro, enteroflavi, rhabdo, orbi, and influenza viruses. They look for a wide range of pathogens, but I can't find something that says we found only H3N8 influenza. Am I reading it wrong? It just seems odd that they found nothing else. However, I'm not a scientist, so maybe I've misunderstood. So I sent Josh's question to Simon Anthony, the first author of the paper, and he promptly replied, Thanks for the question. Your listener is quite right. We did find other viruses in those animals. I have a strong interest in the ecology of co-infection, so was very keen to see what other viruses might be present. We also found herpes viruses and adenoviruses in addition to influenza, though none of these viruses were found in all 5 animals, and none could be linked to the observed pathology. Interestingly, we found 3 different adenoviruses. 2 of them were known seal adenoviruses, and 1 was actually an avian adenovirus. So it would seem that avian influenza was not the only avian viruses that spilled over. In addition, we performed deep sequencing and identified some novel picornaviruses. We're working those up right now, but would love to chat more about them with you at some point. Hope all is well over the road. Yes, Simon is right across the street from me.
Cool. Yay for the adenoviruses.
These seals are messing with birds, huh?
Yeah.
Well, they're going to commonly be in the same places because they're both after the fish, right?
I guess. In the water, right?
Yeah.
known seal adenoviruses. Cool. Yeah, I guess I think they were looking for flu because the idea was that the seals had died of probably flu-like illness, right?
Right.
But then they find other things. If you got to the samples, you might as well look for everything. It doesn't cost very much these days.
That's right.
Okay. All
Good
right.
question.
Excellent question.
Yeah, very good. But Josh, you had it right.
Yep.
But I'm glad you re-listened.
Mm-hmm.
All right, now we have 3 things to discuss, and one of the things is a paper, and the other 2 are outbreaks, which have really not been published as papers yet, but I'm sure will be. And the first one is an outbreak of a hantavirus in Yosemite National Park, which happens to be in California, and not too far from where I just was last week, San Francisco. And this hantavirus has apparently, well, I'll give you the numbers here. 8 confirmed cases of hantavirus pulmonary syndrome and 3 deaths. And these are among people who are staying at what's called Curry Village, which is a bunch of cabins, I think, right?
Tent cabins.
Tent cabins. What's a tent cabin?
These are. It's a wooden platform with a tent, sort of an old school tent pitched on top of it.
Yeah. I've stayed in those with Boy Scouts. Yeah,
Right.
they hate them.
Oh, they're a classic rite of passage for Boy Scouts, I think.
Terrible. I'd rather be right on the ground. The wood is really hard. You put a sleeping bag on the wood. Anyway, what's a hantavirus? Hantaviruses are bunya viruses, and we talked about a bunya from China not too long ago.
Uh-huh.
Right? Envelope negative strand RNA viruses with a genome in 3 pieces, a large and medium and a small genome piece. And the hanta is a genus in the bunyavirus family. And the disease caused by hantavirus was really originated, or our knowledge of it started in the 50s with an outbreak in Korea. There was an illness which eventually ended up being, it was first called Korean hemorrhagic fever, but now is called hemorrhagic fever with renal syndrome, HFRS. FRS is Fellow of the Royal Society.
No.
And if you have an H in front, you're in big trouble. It took about 25 years to figure out that this disease was caused by a hantavirus. And these viruses are typically carried by rodents. And that's the natural reservoir. The rodents seem to be okay, but then it gets into people, and then people get sick. Now, you may remember there was a very famous outbreak of a hantavirus disease in the southwestern United States in the early 90s. And that is a different disease, hantavirus pulmonary syndrome, a lung infection quite severe and often fatal. And that was by a different virus from the one that caused the Korean outbreak. And that was called Sea-Nombre virus.
But it has a name.
And that's after many other possible names.
Yeah, I remember Muerto Canyon.
Right. And it was first thought of as, or known as, the Four Corners virus. But the Native American population did not like having something associated with their area. So in fact, I think there was even Navajo virus.
Yes, it was Navajo virus for a while, and that didn't go over well at all.
Yeah, the Native Americans
flew. That's
what great.
Native Americans objected to Navajo virus. I think the local Chamber of Commerce objected to Four Corners virus because they market themselves as the Four Corners area. Right. And eventually it came down to Sea-Nombre, which I guess is there's a feature, a geographical feature in that area that's Sea-Nombre Canyon or something.
No,
I think it's just the no-name virus.
Or is it just? Is it really just that? Oh, I had assumed that there was some actual location that nobody cared about. Called
it's a plug. It's a
nameless virus.
It's a plug because the CDC got tired of their name being rejected. It's really very cynical. Well, anyway, so it's Sea-Nombre. It's cool.
What else are you going to do?
Yeah, you're supposed to name it over sort of where it came from, like the next virus or the third virus in our story today. Mm-hmm. So Sea-Nombre, the natural host, is the deer mouse, Peromyscus maniculatus. And
cute little guy.
It's very cute. In fact, there's a great page at CDC, rodents in the U.S. that carry hantas. So they tend to be specific rodents and specific hantaviruses.
Right.
So the deer mouse is bad.
They have different ranges, but they overlap.
Yeah. So the deer mouse, as far as I know, isn't very common around here, right, in the Northeast. But in other parts, I have a map somewhere. But
I do too.
Not here. Not here in the Northeast.
Actually, the map that I have it's pretty much all of Maine, New Hampshire, Vermont, New York.
Really?
Not so much
extend
Massachusetts.
down to the coast. Not so much the coast.
Right.
So New York, right down in the city, probably doesn't have deer mice, but upstate you would.
Right.
How about Long Island?
No.
Yeah, because there was one case out there in the 90s, and I think it was a different species, and there was some confusion about it. It wasn't Peromyscus maniculatus. Anyway, there are other kinds of rodents: cotton rats, rice rats, white-footed mouse. That's Peromyscus leucopus. They carry different hantaviruses, but not this fatal C. nombre guy.
Yeah, we probably have the white-footed mouse in this part of the country.
Yeah. So
in fact, I think I've seen those in my shed.
I think I have seen the number 40% of mice in the U.S. carry some kind of hantavirus, but not necessarily C. nombre.
Right.
So I'm saying this because there's been a lot of hysteria over the Yosemite outbreak, but not everyone's going to encounter C. nombre, most likely. So why these people get sick is the I see someone pasted a
that's me.
map.
There you go. There's different.
Oh, there's nothing in New Jersey. I mean, it's very little.
Right? The interesting thing is that in the studies that came out after the Four Corners incident, what fascinated me is that it gave a new relevance to all of the ecological studies that had been done on the distribution of these different mice.
Right.
The mice, by the way, typically harbor this virus with a fairly high frequency, I think, and it doesn't do typical of this kind of thing. Zoonoses doesn't do anything to the mice. They harbor it as a persistent infection, and it doesn't cause them any problems. So there's all of these mice running around, and I think that there are different hantaviruses. I think I'm correct about this. There are different hantaviruses that have a different species specificity for different mice. So there's a fairly wide distribution, not only of the mice, but of the different viruses. But it's only under certain ecological conditions that we can go into when there's really a problem in humans.
Right.
See,
and nobody really much cared which mouse was until all of a sudden we did care.
Yeah, there were just a few ecologists that cared, all right?
Yeah.
And then all of that stuff becomes relevant in a different way. I don't want to imply that it wasn't relevant to start with.
Sure, right.
The original outbreak of HPS in the Four Corners region was associated with a lot of rainfall that year, increased pinon nut crop, the mice coming to eat the pinon nuts. And they go in your house, they urinate and defecate, and the virus is then there. And it dries up and aerosolizes, you inhale it, and you get HPS.
That made the population expand so much that then when they kind of used up all the pinion nuts, then they went into homes.
Yeah, that was my understanding as well. You got a lot of mice who, after a while, are looking for a meal, and it's followed by the dry season where their feces and urine is going to dry out in the houses. So you got mice in the house, and then you go around sweeping up, and you create a bunch of aerosols, and you acquire this stuff in your respiratory tract, and boom, you got a problem.
Right.
And also to that mice in the homes kind of thing, it's really interesting that back in '93, one of the physicians was a Navajo himself, and he interviewed a medicine woman, and the cultural history or whatever had been really very strict against mice. And if your clothing touched mice, you were to burn your clothing. And the whole feeling was that there was something bad in the air. And so it's like over, I don't know how long, hundreds of years, this empirical knowledge of the mice being in the human habitation was a bad thing, was probably something against, infections coming from these mice.
Sure.
The medicine woman said the mouse would choose the strongest and best person in the house, and that reflects on the fact that these hantaviruses seem to be more of a problem for healthy people in the middle age group and not the very young and not the very old. So, it's sort of a mouse taboo that there was some lamenting that as generations had gone by, that recently people had kind of ignored some of these strictures, like, mouse touches your clothes, why do you have to burn your clothes, kind of thing. So,
I participate in a email list among mammalian biologists, and it's very interesting because they have discussion. A lot of them trap mice and study them, and there are these old guys who have trapped 40,000 mice, in the last 50 years. And at one point there was a discussion about what precautions you take. And these guys are like, I don't wear anything. I don't wear no gloves. I don't wear no mask. And I'm fine. I never got sick. So it's pretty funny that, a lot of those mice probably had viruses, but the right combination of situations didn't occur to infect them. Because if I were going out.
People used to mouth pipette acrylamide too, but we know better now.
I used to do that. I used to
do that.
I think it makes you better. Anyway, when I'm trapping mice, so I wear gloves and stuff. So I don't want to get any virus because I'm sure there's more than just hantas. It's interesting to note that this C. nombre apparently does not go person to person. It's never been shown to spread. So it's a real zoonosis in the sense that every infection comes from an animal. So now the thing is that the Yosemite outbreak is probably C. nombre, but I have not seen any sequence anywhere. Have any of you?
No.
I haven't seen a sequence. And up on ProMed, they say it's most likely C. nombre, but and that's based on the disease. So there were mice in these cabins, and there were probably droppings and aerosol virus, and that's why these people get infected.
I haven't seen any real good explanation anywhere as to why this particular outbreak.
Right.
It probably has to do with a higher than normal concentration of mice in these houses in this area, but why that is, I don't know.
Yeah, they had so it's I guess it's still at 8 confirmed cases. 7 of which were people who stayed in these cabins that I guess are a campsite that you drive to, and then one of them was somebody who'd gone hiking up in the mountains, but he had stayed in. I think they have similar types of preset tents up in the I've got it here somewhere. They call them the High Sierra tents or something. High Sierra Loop. Yeah, so he had gone hiking there and stayed in those preset tents. So I think it looks like there was just an unusual concentration of rodents in these tents that are set out, and I don't think anybody's really pointed out why that is.
Is it? I thought it was dry this year out there, right?
It could be.
Maybe. Yeah, I don't know specifically about Yosemite. I know most of the country obviously has had huge drought problems.
Maybe Yosemite was more moist because it is a valley after all.
Yeah.
It could be different. But there's it's something they may try and figure out. So I
have stayed in those tent cabins, and I have hiked this these High Sierra trails, and it's really one of the most beautiful places in the world. Hantaviruses be damned. I'm going.
I think they're all closed for the moment, right?
Yes, they shut down the. I know they shut down the main campsite. I don't know if they shut down the cabins that are in the wilderness, and I don't know what the rules are there on camping in the woods. If you can just pitch a tent somewhere.
No, you'd have to have wilderness permits, and they'd be pretty strictly regulated.
Okay.
I think in
because
Yosemite.
a lot of parks you can just they have just rules. You have to be a certain distance from a trail or a body of water. But this may be because of the amount of traffic they get. They may restrict it to just these preset tents.
Well, or a certain number of wilderness permits right. Yeah, you might be able to free camp, and not just in these Sierra Loop tents.
Right.
Yeah.
So I think they've closed the sites. They're cleaning it up, disinfecting, and they're trying to reduce the mouse population. I read somewhere, but I think that's pretty futile because you can never get rid of all the mice.
Yeah, the problem is you've got these tents that obviously are easy for mice to get in and out of, and you're never going to change that.
Yeah.
and they're rented out to the public, so people are going to go in there, and of course they're going to spill food. And I don't know how the platforms are on these tents, but if they have little cracks like the ones that I camped in as a Boy Scout, then the food's going to go right through, and the mice are going to be underneath.
These are definitely not going to be mouse-proof. No question about that. Actually, this avian flu diary site that you picked out, Vincent, has some really cool figures on it. It's got the hantavirus pulmonary syndrome cases by state of exposure, and it's got the total cases 602 as of July 3rd, 2012. I guess that's 2012 cases. And so, it kind of shows their distribution. And then further down, it has the geographical distribution of 3 different
rodents,
rodents that carry the virus. So that's really nice. And then all this stuff on how to deal with it.
This is a really, really good, we should definitely link to this avian flu diary blog post.
Yeah, that's nice.
It's pretty rare in the end, though. I mean, so, like Rich said, he wants to go camping there. The hell with the mice. So you got to make your decision. But
if the hantavirus chases some people away from Yosemite, that's probably good because it's crowded.
It is really crowded, yeah. CDC says minimize contact with rodents.
Right.
It's funny. We were recording at ICAC. We were in the Moscone Convention Center. And at the end of the day, after everyone had left the room, we were breaking up all the cameras, and a mouse ran across the one of the camera guys said, I wonder if it's a hanta mouse. He wanted to catch it and take a picture, but he couldn't.
Odds are just slightly less than 50% probably if it's here in California and you see a mouse.
Yeah. The hot spot for hanta is really the Southwest. You can see major. But anyway, 600 total cases since we started recording them there, and that's not a lot.
Yeah, and here we're talking about an outbreak that's made national headlines, and it's 8 cases out of everybody who's camped in Yosemite in these tents. I don't know what their traffic is. I don't know what the denominator is here, but this is one of the most popular parks in the country, if not the world. So
yeah. I think early on the CDC was saying 10,000 people at risk because maybe that's the number that camped there.
The denominator, yeah.
But they're just waiting, right? There's a 1 to 3 week incubation period, so depending on when the last person was there, it's probably over by now. The likelihood that we're going to get any more cases, but I don't think people should be afraid. I think they should just, be aware of the risk.
Yeah, you should minimize your contact with mice and probably wild animals in general. And
I'm not getting near you, Alan.
Yeah.
And the symptoms of this is it pretty much kind of starts out with flu-like symptoms, but then really rapidly it deteriorates and the lungs fill up with fluid, and in a fatal case. So there's some concern that, and there certainly was in 93, that would, as flu season approached, would there be more risk of missing these hantavirus infections because they start out looking like flu and might just get dismissed because, that's just the flu.
Yeah. Less severe cases, right? I'm sure, like any infection.
Mm-hmm.
Even Ebola has less severe infections. Okay, that is our first outbreak. The next one is an interesting outbreak of unusual influenza in the Midwest. And I just went to a talk by Lynn Finelli at ICAC. She is a epidemiologist at CDC, and she gave a talk on this. So I'm just ready with all the facts. She was really good. Actually, she also was interviewed by Jeff Fox at the same meeting, and I'm going to put a link to a video of that interview.
I listened to the first part of that. It was good.
Yeah, her talk was really comprehensive. She actually, I have to take an aside here. I moderated the session, and she came up to me before, and she said she got her undergraduate and PhD degrees here at Columbia. All right, she was a undergrad, and then she did a PhD in epidemiology in the School of Public Health. So that's pretty cool. So she must be right.
Yes, absolutely.
Anyway, pigs. Flu is endemic in pig herds all over the world. 50 to 90% seroprevalence. I was amazed by that. And they use vaccines in many farms to prevent illness. From 1931 to the 90s, the only flu virus known to be in pigs was H1N1. And then in 1998, the human H3N2 virus infected pigs. And that's the H3N2, that's the Hong Kong flu that emerged in 1968. And then it began reassorting with the H1N1s that were already in pigs. So you have all sorts of reassortants in pigs, H1N1, H1N2, and H3N2. Starting in about 2005, there were cases in people of H3N2 viruses infecting people. So these are called variant H3N2 because they come out of pigs, and many people don't have antibodies to them because, remember, they went into pigs in the 90s, in 98. So, and then the H3N2 changed quickly after that. So the vaccine is different from that 90s vintage. So from 2005 to 2012, there were 36 cases of human infections. And in those years, in 2007, it became a reportable disease, so it's called novel influenza. In 2011, there were 12 additional cases, and now these viruses are also H3N2, but they have now acquired the M protein gene from the 2009 pandemic H1N1 virus. So it's another reassortment. So the M gene is one of the internal, encodes for one of the internal structural proteins of the virus. Now, most of these cases are associated with agricultural fairs. And she said there are 6 cases that they've labeled as human-human transmission. And she said they have a really broad definition of what that is, and that's no known contact with pigs or pig fairs. So they assume it was transmitted from a person, but she acknowledged that it may not necessarily be transmitted.
How many cases would fit in that description?
So up until 2011, 6.
I see. Okay, we're talking historically. Fine.
Yeah. Historically, right.
Then in 2012, that's this year, that's the big number, 302 so far. Variant flu, again, with this, it's an H3N2 with the M protein gene.
And this is like 20-fold more than usual.
Yeah, it's a lot.
This is a real outbreak.
This is a real spike.
And she showed a map of pig density in the U.S.
The swine belt.
The map she showed was all blue. No, it was mostly blue, but it was really blue in the Midwest, which is where most of the cases have occurred. They've been over 10 different states: Minnesota, Maryland, Pennsylvania, Wisconsin, West Virginia, Illinois, Ohio, Indiana, and even one or a couple in Hawaii. 102 in Ohio and 138 in Indiana.
Well, you got to have pigs in Hawaii for the luau's.
Yeah, I don't know if it was that or imported. So in 2012, that's 10 states, 302 cases, 1 death. And 10 now, of those, 10 they think are human to human by these broad criteria.
And they're mostly kids.
Yeah, and they're mostly kids. It turns out that the kids, if you're below a certain age, you were never infected with this vintage in the 90s, so you don't have antibodies. And very old people tend to be very susceptible. There have been a couple of serosurveys in the U.S. and Norway. Very young and very old individuals don't have antibodies, so they're quite susceptible.
So either you weren't exposed or your immunity is fading, and that's
when. And she said the young kids are the ones that go to the agricultural fairs,
Of course.
And pet the
pigs.
They pet the pigs, or they have pigs and they're presenting them. And she said, a lot of these pigs they've raised for a while and they stay with them in the pig house because they don't want to leave them, They sleep next to them and that sort of thing. So if you're a pig handler, you have high risk. If you're just going to a state fair, the risk is much lower. But she said there are some cases where people just go and they look at pigs and they touch the wood on the fence or whatever and they get infected that way.
These are prize pigs. You can pay your college tuition with one of these pigs.
Yeah.
Oh, they're worth some money, yeah.
So let's see, here's some cool numbers. So they have data for 203 cases. 198 had direct or indirect swine contact. And 106 of those had pig exposure for 2 to 7 days. And this is a cool number. 50 million people have attended agricultural fairs in 2011. And 30% of those visit the swine barn. So her predicted number of cases based on what's occurred so far is 500 to 900 by the end of 2012. But she said, the weather's getting cooler, and that could make transmission better, because flu transmits really well when it's cool and not humid.
Well, but the agricultural fairs are mostly ending.
Yeah, so that's
I think
good.
we have one of the last ones in the country here in Massachusetts, right up the road from me, is the Big E. So that'll be happening. I think that's starting maybe this weekend.
Why don't you go check out the pigs, Alan?
Go to the pig barn.
Go check out the pigs.
Go to the pig barn, yeah.
Up.
So you, let's see, you're probably immune to this because you're not old and not really young, as far as I know.
I certainly was around in the 90s when this thing was circulating.
Now the one person that died was an adult who apparently had some real serious predisposing conditions.
Yeah. She said the people with bad health for various reasons get the most sick, as with with flu.
The people are priority for getting flu vaccine.
Yeah. So this is interesting. The CDC has provided 2 vaccine candidates to manufacturers in case they need to be, if there's an outbreak and they need to be manufactured and distributed. And these are the usual, there's egg or cell-grown reassortants, high-yielding reassortants with the right hemagglutinin. So currently, our flu vaccine has an H3N2 component,
right? But
it doesn't match this pig virus. It's quite different because, again, the pig H3N2 was circulating in the 90s. So a writer asked a question. This is Maren McKenna, who writes for Wired magazine. She said, "Are you worried about this virus getting into the massive pig farms that are in this area?" So what we're talking about here are agricultural fairs. Those are, right, locally raised pigs.
By the way, this virus isn't doing anything to the pigs, is it?
And she said the pigs are usually okay. Yeah, they don't really show symptoms.
Okay.
I suppose if they did, you wouldn't bring them to the fair, right? Although,
not.
This is not my area, so I don't know.
Yeah, you wouldn't exhibit a sick pig.
Yeah. So Finelli said that it's really hard to... There are a lot of biosecurity precautions taken on pig farms nowadays. It would be really hard for... you wouldn't get one of these pigs in there. You can't bring anything in, and the people have to be treated in certain ways as they go out. So she thought it would be unlikely that a virus would be brought in. I don't know. Could a worker be sick with one and bring it in, though? Who knows, right? Could happen. Yeah. So anyway, that's... so the last thing I wanted to say is that this M gene, a lot of people have said maybe the M gene is helping it transmit among people, right? That's why it was selected for 2009 H1N1. But a paper came out earlier this year where they compared the aerosol transmission in ferrets of these H3N2 from swine with or without the 2009 H1N1 M gene, and the transmission rate is the same in ferrets.
And ferrets are a perfect model of humans.
Yeah, exactly. So we can't have it both ways, right?
No.
So I'm saying that doesn't mean very much for people, but I just thought you might like to know that.
Right.
And
so the other thing I like is this MMWR report that you found, Vincent, that apparently the CDC was motivated to go reassess the rapid diagnostic flu tests.
Yes.
In the wake of this. And so there's the rapid diagnostic tests, I guess, are things that doctors can use in their office, and they're dipsticks of some sort or another. I've never seen one of these that you can put nasal wash on, and they detect influenza antigens. And I must say that it looks like they weren't really very good. They were, about 50% accurate, whereas the CDC uses a PCR test that's quite accurate. So it said, yeah, sure, go ahead, get the test, but don't trust it. If you're suspicious, send us the samples anyway.
Yeah, that's about the number I've heard the past couple of years, 50% accuracy. So if it's negative, it's not necessarily negative.
Right.
But if it's positive, is it positive? Because they're only reporting the negative failures.
Yeah,
here,
I
you know?
don't know.
I didn't see anything about false positives. Yeah.
I don't know. The one question I have is why there's this big spike this year. 300 cases.
Well, I wonder if it's, the pigs themselves, right? Is there a much higher incidence in the pigs?
I don't know.
Somebody going to go out and assay these pigs?
Well, they do have a pretty good surveillance, but of course it doesn't get all pigs.
Or maybe the virus finally varied enough and, I don't know, made it more transmissible?
It does better in humans than before?
Epidemiologically, there's very little evidence for human transmission.
Right. I'm saying to get from pig
to human.
Humans, there's a certain amount of ability to replicate in humans that's required, and maybe it has just passed a threshold and now it transmits more easily from the pig to the human.
So Lynn Finelli didn't address that?
No, but to do that, you need serial passage in people, right? From pig to human over and over is not going to always select for the same improved trait. So,
right.
Unless it went from a pig to a person and then back into the pig, and in that pass, it got something that helps it among people, go to people, and then spread in the pigs. I mean, we're just speculating, but presumably they sequence a bunch of these, they'll start to get some clues. But they're saying we have to keep an eye on this because this could cause an outbreak. There are enough susceptible young and old people that it could spread. So.
Or it might be that we've crossed demographic threshold, where you have enough kids now that are potentially susceptible that you're actually getting a spike in cases. It's just speculation.
So pigs are very interesting. I mean, viruses can go into pigs for many years, and they don't change much. So that's the story with the 2009 H1N1. That had been in pigs for ages, and it didn't change. And then we lost our major population immunity against it, and that's why it was a pandemic in 2009.
Right.
So this H3N2 could hang in pigs for 20 years, and then boom, not much H3N2 immunity, and it comes out. Or could be in this year. Who knows? So we need a universal flu vaccine.
Yes, we do.
Yes.
I don't have to worry about this.
I know all the major manufacturers are working on it.
Yeah. Yeah, there were a
and
few
a lot
talks,
of basic researchers too. So.
I'm sure it'll happen. All right, our next paper is about Heartland virus. I like that name.
Yes.
It's the New England Journal of Medicine brief report. It's a pretty comprehensive paper. It's brief report is.
I really like this. I love these case-based papers.
This is from a big collaboration, CDC, local health people in Missouri, and Stuart Nichol, who was at the CDC.
Who, in fact, was a major player, probably the prime mover in the whole Four Corners hantavirus thing.
That's right. That is right.
He told me his, at least to that point, his biggest scientific high was at about 3 or 4 in the morning when they developed the gel using primers for some hantavirus, and they got a positive signal on the gel with the Four Corners isolates. So they already had some antibody evidence then that suggested that what they were dealing with was a hantavirus, and so they were using these primers, and he said, it was really cool to get that result.
Yeah.
But did they fulfill Koch's postulates?
Well,
for Hanta.
Yeah.
I don't think you can anymore.
Yeah.
Interestingly, that, of course, Hantas are bunyaviruses, and so is this new virus that he discovered. Bunyaviruses. This is a different genus, though.
Right, in the Flebo genus.
Flebovirus, which is a good name. I always like that.
Yeah.
Go to a phlebotomist or a Flebovirus. It's 2 case reports, 2 guys who live on farms in Missouri, but they are not the same farm. But they're in northwestern Missouri, and they both had a history of tick bites.
Yep.
One guy had 1 tick on his abdomen, and the next day he got fever and then developed nausea, diarrhea, and ended up in the hospital. Pretty sick, 10 days in the hospital.
Yeah.
But the other guy...
This is...
I can't
just dropped my jaw.
He had an average of 20 tick bites a day for 2 weeks. What is he doing?
Yeah.
All right, somebody tell me. So if you have like wheat or something or corn, are there ticks in there?
I don't know.
Yeah, I don't either.
Kathy's the one who lives in the Midwest.
Yeah, but I've never been.
You don't get 20 tick bites a day, Kathy?
No. And you can use repellents, I think, for ticks, right?
Sure. Yeah.
Yeah.
So,
And if I lived in an area that had, I mean, I go hiking for a day and I come home and everybody in the family checks themselves for ticks.
yeah. Well, and he checked, and there were 20 a day.
20 a day for 2 weeks.
But he actually got bitten.
Yeah, they were embedded.
Yeah, they've got to be on for at least a little while before they bite. And
you're supposed to take precautions. Long sleeve shirts and pants. Tuck the pants into your socks. But I guess
spray some permethrin on your clothes. Yeah.
Anyway, so they both got sick. This is the second guy within a week. The last bite before getting sick.
Guy, patient 2 with the tick bites, is a 67-year-old guy, okay, who is a farmer in Missouri. He's not,
he's not going to take
rolling up
precautions.
his sleeves, dude.
Yeah, okay.
Going out there,
right.
okay?
If you told him to take precautions, he'd laugh at you and walk away.
Yeah, he'd laugh at you. He said, I'm 67 years old. I've been doing this all my life. Come on, give me a break.
Yeah, they're both old guys. With 67 and 57. So they've been doing this a while. It's very interesting. So they're both really sick. They have thrombocytopenia. Remember that from that paper in the China case? You guys maybe not remember.
I decided I wasn't going to review all that.
It's okay. You don't have to. But I'm just saying, we talked about a Bunya infection in people in China in rural areas. It seemed to be tick-borne, and one of the symptoms was thrombocytopenia.
Decrease of platelets in the blood.
Yeah.
Yep. Yeah, in fact, I think that was the red flag here, because otherwise these guys show up, they've got your basic viral prodrome flu-like type of stuff. I think they both had diarrhea, or one of them did. Mm-hmm. But other than that, it's just, fever, malaise, fatigue, yada, yada, yada. But they had this thrombocytopenia, and the blood work suggested something else is going on here.
Yeah, the Chinese Bunya virus was called severe fever and thrombocytopenia virus, something like that.
Right. Yeah, so these guys didn't just have the flu.
And they look for rickettsia, which are borne by ticks, or Ehrlichia.
Right. And rickettsia is associated with many of these same things: leukopenia, thrombocytopenia, hyponatremia. So
they give antibiotics, and they probably just got better on their own, right?
Yeah.
But they said it's a good example of how these guys didn't need antibiotics,
Right, but you didn't know that until kind of late in the game.
Didn't know it.
Part of it is if you give the antibiotics and there isn't any improvement from the antibiotics, then you start suspecting that it's something else.
Right. No, I'm just saying
they placed empirically on doxycycline means we're going to see if this fixes anything.
One guy got a 14-day course, right?
Right.
I mean, I'm just talking about it in terms of antibiotic stewardship. You really need to be careful. Of course, if you don't know what it is, then you have to take some chances. Otherwise, you'll lose the patient.
Yeah, these cases
they're tough.
Are the type of outliers where you'd say, well, yeah, antibiotic stewardship, but you've got to try some stuff on these guys, and they're kind of aiming at the stuff that it's likely to be.
What we need is a tricorder to diagnose these cases.
Of course.
And
you were just talking about that rapid flu diagnostic, which is it's that technology, but it's not accurate enough yet.
So here we have this week in vocabulary. When you start a therapy before a specific diagnosis, it's called empiric therapy.
Okay.
Right.
Empiric therapy. Yeah, I mean, I hang around with microbiologists now, and they say, you get certain symptoms, you give a broad spectrum antibiotic until you get the lab results back. Right. So sometimes you'll know right away that there's sepsis, but you don't know what the agent is, so you treat broadly, and then you can focus it in later.
Yeah, because if you wait until the lab results come back for everything, then you get a lot of dead patients.
Yeah. By the way, speaking of tricorders, one of the keynotes at ICAC, the guy, his talk was about diagnostic methods. And he said, in 10 years, we're going to have a tricorder, some kind of external sensor that can diagnose a lot of conditions. And in fact, Qualcomm has a contest on at the moment. It's a $10 million prize to develop a tricorder that's less than 5 pounds, and within 5 minutes will diagnose about 10 different afflictions.
Oh.
So, Rich, you better get working on it. You can get your 10 million.
Yeah.
Anyway,
And just in case somebody doesn't know, the tricorder is something that comes from Star Trek.
that's right. Did you know that, Kathy?
I suspected it.
But you don't watch Star Trek, right?
I had watched Star Trek and some of the movies, but
Yeah.
I'm not a Trekkie.
Yeah, in fact, this speaker showed a picture of Spock with a tricorder. It's funny.
Yeah, it's one of the images that comes up right away on Google.
If you search for tricorder, really?
Yeah.
That's great.
So, just as an aside, platelets are really cool. They're these basically cell fragments spun off from a megakaryocyte during hematopoiesis. So they're part of the whole blood lineage, but they're cell fragments. They don't have a nucleus, and they initiate clotting when there's injury in blood vessels and elsewhere. Even though they don't have a nucleus, they've got all of these sensing systems on them, and they can sense damage and go sit on it and literally sit down and plug up the hole and call in all sorts of other cells that are going to help build a clot. They're amazing.
It will be interesting to find out why this virus causes that, right? Maybe it's infecting bone marrow and causing a defect in the production of the precursors,
Well, and this is another zoonosis, right? So this,
presumably,
as far as we know, this virus is not really evolved to be in people. So, I mean, I don't know what ticks get. Typically, these viruses, by the way, are insect, or ticks aren't insects, right?
Right, they're
Arthrovirus.
Are
Arthropods.
born by arthropod born. Okay.
Yes.
So, I don't know. Do ticks get thrombocytopenia?
Not hardly. They don't really have blood like that.
Do they have platelets?
I suspect the ticks are okay.
Yeah.
So these guys were sick for... Actually, this happened 2 years ago.
2009. Yeah.
Yeah, it's a while ago. They got better on their own, basically. What is it, 4 to 6 weeks? They got better, and in 2 years they've been fine.
They were really sick.
They were really sick.
Yeah.
One of them
had
still
some
complaints of headaches and stuff or something.
Yeah, but they can't be certain that that's due to this. But one of them does seem to have possibly some sequelae from it.
Now, they took white blood cells, leukocytes, from both these guys. They inoculated them onto macrophage cells, cultures, and they developed CPE, and they took the supernatant and passed it, and those developed CPE, and they could find virus particles in them by EM.
They wound up infecting vero cells even, and it grew
Yeah.
on them.
Yeah.
I wonder if this was done while these guys were in the hospital. It doesn't really give the timing, right?
I'm thinking that this all evolved sometime after that, because, like you say, it was 2 years ago, the cases.
Yeah.
Right.
Right. This the second author on the paper, Scott Folk, is the MD, and I did see somewhere that the CDC pays attention when he sends them something. And so, yeah, you couldn't quite tell the chronology of it, but I think basically he sent them a sample from second day blood draw, from second day in the hospital.
Huh.
So whether he drew it and saved it, or when he sent it relative to figuring out that the antibiotics weren't doing anything.
Yeah.
Maybe.
Yeah, I gather he has a reputation as a pretty sharp diagnostician.
Right.
So it's not just, he's sending us this because he hasn't run the right tests. It's hey, this might really be something in here.
Mm-hmm.
What I found interesting was if this chronology is the order in which they present the results, they go to EM, and then they go and they isolate RNA and try and find sequence.
Mm-hmm.
Yeah, if that is in fact the sequence, who knows?
If that's the sequence of events.
Yeah, it could have happened at the same time.
This electron micrograph is
beautiful.
really cool.
It's very nice.
Yeah.
It's
really
amazing.
cool.
Yeah, it's you look at it and you immediately see what's going on.
Yeah.
So they got RNA from infected culture media. Not from tissues. From infected cells, and they sequenced it, and they found that this was a novel bunyavirus in the phlebivirus genus. And quite different from most others, except very similar to this severe fever and thrombocytopenia virus that was identified in China not too long ago.
And I gather, I guess CDC then went into the field and tried to find it.
And they didn't.
So they went out collecting ticks, which I guess at least at this one guy's farm should have been pretty easy.
That's right. They made you
they found
A. Americanum, right?
Yeah, they weren't able to find any that were carrying this virus.
Ambyloma americanum, the most common tick in the area.
Right.
Southern Missouri. I like this one statement. This just evokes something. The novel virus was also distinct from an uncharacterized Bunya virus called Lone Star virus, which was isolated in 1967 from a tick found on a woodchuck in western Kentucky. I just have this vision of a guy walking around and he finds this woodchuck and he picks a tick off and it happens to have a virus. I know it's not how it worked, but the statement is evocative.
Somebody happened to be looking there for some reason and they found that, but this is not that virus.
Yeah.
Right.
They find viral RNA in bone marrow from one patient. They had actually done a bone marrow tap on the guy because I guess they couldn't figure out what was going on, right? They wanted to know.
Yeah.
And they got antigen in the bone marrow cells as well. And then both guys turned out to have antibodies, high titers of antibodies to one of the viral proteins.
2 years later.
Yeah. So, did this virus cause their illness?
Probably.
Yeah.
But as the authors carefully note, they have not fulfilled Koch's postulates.
Right. I'm not volunteering for that experiment.
No.
I mean,
there's just too much coincidence here between, the ticks and the type of virus and the symptoms that were caused and everything else. It walks like a duck.
Yeah. I mean, one of the strongest pieces of data is the serology. All right, so they did ELISA with these guys' sera using inactivated virus. Okay.
Yeah.
So there's no specificity control there. Is there any cross-reactivity with others, right? I'd like to see that because if there maybe a different Bunya could cause this ELISA positivity.
Yeah. But these guys both had tick bites. This is a common MO for Bunya viruses. They developed a Bunya virus-like syndrome. You got Bunya viruses isolated out of their blood samples, and you go back and they've got antibodies against this Bunya virus. I would agree with Rich that, this is about as close as you're going to get to.
All right, I'm going to play devil's advocate. If this was XMRV, you would say no.
XMRV didn't present this way.
I know, but you had, X cases with PCR positivity and X controls.
But that's very different.
How's it different? They took blood from these people. It's a very different case
because you're looking statistically at two populations of people and saying it's more common in population A than population B. In this case, you've got two people who showed up at death's door and unrelated cases who showed up, and you drew blood from them and you isolated the virus directly from their blood. I think it's a lot clearer.
I'm not sure. I mean, the other case, you had people with chronic fatigue syndrome, isolated over various times and in different places, and they had blood from them, and they pulled out this virus. I'm not saying that. I'm just saying that let's apply a strict standard to all isolates of new viruses.
Yeah,
right?
and I think you can apply a strict standard that would include this study as probably causal and would exclude the XMRV studies as evidence of causality.
What if it turns out that this is a contaminant?
It could be.
It could be.
But given
they
the
actually
assays,
grew it. Well, it would have to.
Yeah,
it's a novel. Look, it's a novel virus, but so was XMRV, right?
Besides the presentation of it, and all the whole fact pattern lining up, you've got the ability to isolate the virus from the patients. You've got a set of assays that is very straightforward, and the serology is solid. in XMRV, you had the serology results that were kind of, and you had these assays that were, we're going to culture it and then passage it and then culture it and then passage it and then culture it and then do multiplex PCR 5 ways from Sunday, and then we see it. And that's a very different ballgame.
I bring it up because some people who had an interest in the XMRV story might be listening and wondering why we're so quick to accept this. I don't think it's a given that these are the causative viruses. I think we need more data. More infections would help. The China SFTSV infection has now been confirmed in a number of patients. So I would like to see that here.
You make an interesting point. It would be nice to have some sort of rubric that could be applied.
Yes.
To globally to
other than
indicate causality for something like that.
But would fall short of having to inoculate people.
Yeah, absolutely.
Yeah, clearly we have trouble with Koch's postulates. There's got to. How about Racaniello's postulates?
No, other people
working on that.
No. Other people have proposed Koch's postulates for the 21st century.
Okay.
In fact, I had a conversation with Ian Lipkin last week, and this came up, and he says we need to have some rubric to identify, to discover new causative agents, and we don't have it yet. And so he speculates on that, and that'll be up next Tuesday.
And in fact, I would say if some of these authors suddenly decided that they were going to go out treating patients with tick bites with some sort of antiviral compound on the theory that this is causing all their ills, I would be very, very uncomfortable with that.
Yeah.
You actually wrote a blog on this, Vincent.
On what?
Koch's postulates for the 21st century.
Yeah, I did. It was an interesting issue. It's a while ago.
Yeah, it is something that's going to come up more and more as the assays get more sensitive as we look more thoroughly for these sorts of things.
Yeah.
And in this case, certainly it would have helped a lot if they could have isolated it from ticks. But there we are. It's a report that I think it probably caused the illness in these 2 patients. And it's something that hopefully people will be looking for now, and the CDC obviously now has assays for it, and maybe we'll find it.
So where did this thing come from, and how much of it is out there?
How long has it been out there?
Right.
Yeah, but there are lots of new viruses for which we have similar questions, and
sure,
yeah.
I mean, these may be the last 2 cases you ever see. Who knows?
Right.
Or you might, but we'll keep an eye on it. We'll see. I think
it's a testimony to the fact that we're getting really good at tracking these things down. There's a lot of these things. Well, I think for years there was there still is ARDS, acute respiratory distress syndrome, right? Which is people who get a respiratory disease and die, and it's never diagnosed what it is. And I think there's a fair percentage of those that have been historically hantavirus, and there's the rest of them are probably something else. We're getting better at tracking these things down and identifying what they are.
Yeah, right. There are a lot of people who show up at a hospital with severe illness, even acute onset stuff like this, and they're treated and they get better or they don't, and nobody ever knows what caused it.
Right.
Yeah.
Right.
Well, speaking of XMRV,
oh dear.
The Lipkin coordinated study will be published next Tuesday. Mm-hmm. And there will actually be a press conference here that Ian Lipkin is running with a number of the major players, Ruschetti, Mikovits, Harvey Alter. So we'll put a link to that in the show notes where it's going to be live streamed on the internet. But that'll be on Tuesday. And Rich and Alan and I will be busy on Tuesday, so we won't be able to watch it. But I did talk to Ian last week and recorded our conversation, and I will put that up on Tuesday on TWiV as a little special. A TWiV special. We can't number it, otherwise it would be 200.
TWiV 199.5?
Yeah, we actually have had it. This is today, and technically the 200th thing, because I did a special in 2009 with Peter Palese, but I didn't number it. But we're only talking about numbered episodes. They're different. Specials are different. They're just usually short.
It's you talking to somebody. It's going to be a shorter than usual type of thing.
Right.
Yeah, so the embargo on the paper lifts at 12:01 a.m. on the 18th, and that's when that'll go up.
It would be a TWiV. This in virology.
This.
Forget the weeks.
Yeah, this. Yeah, that's good. I like this in. Okay. By the way, Scott had sent me a link to this paper a while ago. Dear Vince, I'm a fan of your podcast and thought you might be interested in the article if you haven't heard of it yet. So that's Scotty sent a link to the New England Journal article and a news piece at Yahoo. Heartland virus. Do you like that name?
It was named after the medical facility where Scott Folk works.
Yep.
What's the name of that? The Heartland Hospital or something?
Heartland Regional Medical Center in St. Joseph, Missouri.
Neat.
Which is pretty much in the northwest corner of Missouri.
But
That's
what
why
is
it showed up.
Kathy, what is the Heartland?
What is the Heartland? It's probably whatever you define it. I mean, what is the Midwest? That's also whatever you define it.
It's the swine belt.
Okay. All right, let's do a few emails.
So should the flyover now be the tickover?
Mm.
No.
Let's do. Let's see. How many have we have here? We have Mark, Justin, and Victor. Okay. Alan, take Mark's email, please.
Sure. Mark writes, could you please discuss the relationship between HPV vaccine and GB? He means Guillain-Barré, I assume, not Great Britain. Also, it would be interesting to know what causes GB and its relationship to polyradicular neuritis, coonhound paralysis, and MS. Do these diseases have any viral relationship? Thanks. I listen to the show while gardening. And Vincent's provided a couple of links. The relationship between HPV vaccine and Guillain-Barré is that there isn't any relationship, as far as I know. And in fact, the CDC, ever since the swine flu, in 1970? 76 vaccine that was associated with Guillain-Barré, there's been a very, very diligent screening and testing and surveillance for that as a possible vaccine adverse event. So the CDC watches for this anytime somebody introduces a new vaccine, and they watch for it every year with the flu vaccine, and they've watched for it with the HPV vaccine, and as far as I know, it has not happened. This is a very safe vaccine.
Quite, yeah.
Guillain-Barré, by the way, is a usually transient neurologic disorder.
Right.
That actually quite frequently shows up following an infection of some sort or another, as if infection can trigger this in some fashion. And it is an autoimmune disorder, which means that your immune system is attacking your nerves. And there are lots of theories of autoimmunity that say that it could be that when you get infected by something and mount an immune response to it, that you can sometimes get a cross-reactivity to something that is part of you rather than the infecting microorganism. So that all fits together, but there's no evidence. So you can create a story where a vaccine could cause something like this based on all of that, but there's no evidence that that's actually what's going on here.
Right, and the comment, it would be interesting to know what causes Guillain-Barré. Yeah, it would be very interesting to know what causes Guillain-Barré and its possible relationship to these other neurological syndromes. I know a little bit tangential, but kind of related, the standard mouse model for MS, I believe it is the EAE mouse, right?
Mm-hmm. Mm-hmm.
And in that model, what you do is you take a perfectly healthy mouse and you inject them with something that causes an autoimmune response against their myelin on their nerves, and it gives them MS-like symptoms. So you can recapitulate some aspects of disease pathology just with an autoimmune response against the tissue. What that has to do with viral infection, that's still an area of active research.
Yeah, the coonhound paralysis seems like another autoimmune disease.
Right.
Creeping paralysis due to acute inflammation of nerves. But I'm not aware of any connection, but it sounds similar to Guillain-Barré.
Yeah.
Guillain-Barré was a side effect of the 1976 swine flu vaccine.
Right.
Nobody knows why, and it hasn't happened with other flu vaccines since then.
Right.
Now, there was a lot of controversy about whether or not that was statistically significant. Is that resolved, Vincent?
I think there was a slight increase in Guillain-Barré over the background, but very, very slight.
Mm-hmm.
So it's just, and it's never happened again, so I don't know what to say.
Yeah. But based on the possibility that they might have been connected, I would agree that the causality there is dubious. But just based on the possibility that might have been the case, you certainly don't want to give people a vaccine that's going to cause a neurological problem, and that has been a standard part of vaccine monitoring and testing ever since.
Mm-hmm.
Right.
The U.S., 3,000 to 6,000 people every year develop Guillain-Barré, whether or not they have received a vaccination.
Right.
And so there was CDC says there was a slight risk of Guillain-Barré in 76 with the 76 flu vaccine. And people were worried that the 2009, swine flu vaccine would have a similar risk, but it wasn't realized.
But that vaccine was also manufactured differently.
Yeah.
But yeah, when that vaccine was introduced, it followed the same surveillance system.
Yeah.
That's one of the little boxes they look to see,
Yeah.
Does it cause Guillain-Barré?
Right.
Does it do any of the other things that we've got on our list? And
so, Rich, in the U.S., the rate of Guillain-Barré is 1 to 2 cases per 100,000 people. In 1976, it went up 1 case per 100,000 people. So that's just too close, right?
Right.
Yeah.
That's
Well,
the
you
problem.
could say it doubled.
Yeah, I guess you could. I think
but that was perceived to be greater than the statistical noise in the in the measurement.
Right.
Yeah.
Better be safe than sorry.
Exactly.
Right.
Kathy, could you take the next one?
This is Justin.
Justin.
Hey, TWiV team. I've been listening to your podcast for only a few months, but listen with rapt attention on my way to work each morning. I was shown the podcast by my professor in my intro virology class during my last semester of my undergrad in cell and molecular biology. I've recently graduated and decided not to immediately re-enter academia for several reasons. The first being I have no idea what type of degree I'm going to pursue, MD, PhD, or hopefully both. Also, and largely, I'm training as an elite trampolinist with my sights set on the 2016 Olympics in Rio. Unfortunately, that means I plan to put the vast amount of my focus and effort in that venue of my life for the next 4 years, and after I retire, I would like to find a school with a good program without worrying where I will be training. Since that could put me out of school until 2016, I'm terrified I will lose my edge for applying for school when the time comes. I'm trying to stay sharp with your podcasts and reading articles that pique my interest, but I don't think it will be enough. My most recent idea is to take the GRE this year and then apply for a master's program in either the biochemistry department or the biology department, which includes cell biology, molecular biology, immunology, and virology research areas at a local university. I flip-flop between the two departments because I've heard on your show the importance of a chemistry education for virology, and that my research advisor previously advised me to not educate myself in solely one discipline. My questions to the TWiV team are whether this is a good plan considering my goals, and which department you would most recommend for me. Thank you so much, and keep putting out the viral podcasts. So I thought it was interesting that as a trampolinist, he said he flip-flops between the department.
Yes.
So a department in biochemistry or biology, master's, I think, in either one of them, if that's what he wants to do, would be fine. It all depends what you pick within those departments, too, and at least what you'll be doing for the next couple of years, but then that doesn't necessarily determine what you would do if you went on to get a PhD or an MD PhD in the future. So whatever you want to do to keep educating, I think is a good plan.
So he basically wants to do this while he's training, right? So,
right.
So he stays intellectually sharp.
Right.
So would this master's program involve research?
It might
could, right?
involve research, but there are master's programs that don't involve research. There are several here at the University of Michigan, and there are some that involve research and you don't have to do a thesis, but there are some where they involve research and you can do a thesis. So at least in our case, in our department's master's, there's all three options.
It seems to me it would fit well so he could put a lot of time into training, right?
Right.
You don't have the pressure of a PhD.
Right.
Or MD PhD.
You're not going to be able to train for the Olympics and do a PhD, that's for sure.
Yeah.
No.
Okay. But I also, you can't pass up a chance to go to the Olympics by any stretch of the imagination. And I would, he's not going to lose his edge. He'll be fine. Go for it. And as a matter of fact, in particular on a med school application, well, all kinds of applications, that sort of peripheral interest really gets people's attention.
And, well, because especially when you do it at that level, if you're competitive for the Olympic team, and that's why you took a couple of years off, that indicates that you're the kind of person who finishes what they start.
Right. Motivation is everything.
That indicates a work ethic that is going to do fine.
Right. Exactly.
And he says, P.S., make sure you watch trampoline in the Olympics. We can reach heights of almost 30 feet. Or get excited and take a peek on YouTube by searching Olympic trampoline. So I did do that, and the Olympic trampoline, the YouTube that I saw was pretty impressive.
I was up in Rochester one day, and I watched some of the trampoline. It's amazing how high they go.
Yeah.
And if you make like a little misstep, you're out.
Right.
Yeah.
I mean, I saw one guy from the U.S. who was highly favored, and he just screwed up. He stepped off, and that was it.
Yeah.
Man.
It's like I looked at it, and it looked like diving, but in reverse. Because they've got to do all these fantastic maneuvers and everything, and the height,
Right. Defying gravity.
Yeah.
I did a lot of trampoline in high school.
Yeah.
But I didn't go 30 feet up.
No. While we were watching it, I said, what do these people do after they, finish the Olympics? And someone said, well, they go work for Cirque du Soleil, and they have great trampolinists. I saw them once, these incredible trampoline routines. So cool. All right, we have one more. It's for you.
But I guess anything you do after being an Olympic trampolinist is going to be a letdown.
Yep.
Yeah.
Is that a joke?
It could be.
Okay. Rich, can you finish up the last email?
All right. I'm going to have to look at the trampoline later, I guess.
What, are you trying to multitask, man?
Yes. Speaking of which, greeting TWiV. In episode 191, the subject of working smart and not long came up. When Rich mentioned multitasking, I couldn't help but think of the Lifehacker article on the subject. It goes on to explain why multitasking, as most people do it, reduces your overall productivity. It is a great read, and it's changing the way I work. And he gives another article on the same subject. And basically, both of these things say that multitasking is, if anything, sort of slows you down rather than speeds you up. And, I get it. I think there's multitasking and there's multitasking.
Yes.
The kind of multitasking I'm talking about is you go into the laboratory. This is all a game. Efficiency is a game for me, okay? You go into the laboratory and you start an experiment that has, an hour incubation. What do you do for that hour? You do something else. You don't sit there and read the funny papers or something like that. And in fact, you can have 2 or 3 things cooking at once, and it becomes a game as to which you should start first, what's the rate limiting step in your day, and all of this kind of stuff, so that you can interweave several tasks where you might otherwise just be sitting around doing nothing, and get some efficiency out of your day. That, as opposed to while you're trying to write a protocol, sitting there with your iPhone and reading every email that comes in. That's different. That's
Right.
not gonna work.
Right.
All right.
Yes, my interpretation of your comment was that you were talking about you start this, and then while that's cooking, you go do this, and so on.
Right.
Yeah.
So-
Or as a colleague of mine puts it, doing your experiments in parallel rather
Yes.
than serially.
Yes.
So
Right.
The way you would cook a big meal in the kitchen.
Right.
Right.
Right. Nevertheless, the point about, I think that this is an important point about a lot of people tout multitasking as if, having 3 cell phones and a beeper and
Right.
all this stuff going on at once is a good thing. And I think that there's a lot of evidence that no, it's not. It actually does slow you down. And that's an excellent point.
Yeah.
It's whatever works for you. Some people can do it and they're productive, and other people can't. I don't think there's a rule for everyone.
Well, but the human brain doesn't actually multitask. You're doing one thing at a time. You're just switching back and forth between.
That's right.
Depending on what the things are, they're either going to be distractions or not.
I was sitting behind a reporter at a meeting, and she was on her computer, on her cell phone, and on her iPad all at the same time, just going between the three of them.
Yeah.
It's pretty funny.
Holy cow, these guys really do jump. Speaking of multitasking.
Look at this guy. Yeah, they do. They really go up. Okay. Hey, Rich, can we do some picks?
Sure.
Or do you want to watch those videos?
No. Okay. We'll do some picks now. That's okay.
Let's see. Let's let Kathy go first, because you should go later.
Okay, I picked a YouTube video that's Euler's disk, and at the beginning of the video, they have a link to the supplier of this Euler's disk, and really a lot of other kind of cool science teaching aids or toys that I thought listeners might be interested in, particularly if they're science teachers or homeschoolers or just the kind of people that like to play with chemistry sets or something like that. But anyway, the Euler's disk is a really cool disk, and it spins. You might find the sound a little annoying, so you can put your computer on mute, but it spins for a really long time. If you go to Wikipedia, they explain the whole physics, and there's equations and scary stuff like that about how Euler's disk works. But I thought it was kind of cool. YouTube, and then link to another site.
These are cool. Yeah.
I looked at this thing. I need to buy this for my grandchildren. It's very cool.
Mm-hmm.
Nice. I would have called it Euler, but you're right. Nice. Alan, what have you got?
I have a video that is on Vimeo, so if you click the link, it's okay. It won't autoplay. It's called Insane in the Chromatophores. So this is these folks are neuroscientists, and they were working on squids, which have these structures in their skin called chromatophores. The chromatophores allow the squid to change color, so it can hide from predators or send warning signals or what have you. And they were fascinated that when they stimulate nerves in the squid, if you detach a squid tentacle or fin and you stimulate these things, you can get the chromatophores to change color because they respond to nervous impulses. Well, of course, a nerve impulse is just an electrical impulse. So what they did, and I thought this was brilliant, was they hooked up an iPod playing a rap video called Insane in the Brain. And they hooked it up to this disembodied squid fin, and the chromatophores pulse in time to the lyrics and the beat of the music. It's like the visualizer in iTunes, only better.
That's cool.
It's fun.
Great.
And then there's, I guess, a link to a TED Talk about this, too.
Yes,
Yeah.
So it looks
apparently
cool.
they did a TED Talk.
Nice.
Neat.
Rich.
Right. So I ran across this in, I guess, our local paper that directed me to this. I had not seen it before. It's the Golden Goose Awards.
Yes.
So the original article is in Science Insider, and that's the link. This particular article says that, the Golden Goose Awards are a comeback to what William Proxmire used to do, which was to give out the Golden Fleece Award in Congress on a regular basis, where he identified some research project that had what seemed to be a silly title and then pounced all over it and said, aren't we wasting all of this federal money on this ridiculous research? And a lot of it had to do with basically complete misunderstanding of the research projects themselves and everything else. So apparently this Representative Jim Cooper from Tennessee helped hatch this idea of the Golden Goose Award, where what he wants to do is, in collaboration with the AAAS, give on a regular basis awards for scientists who, just sort of messing around, came up with something really cool.
Basic research that laid a golden egg.
That's right. Basic research that laid a golden egg. And apparently the first one is going to go to Martin Chalfie for his discovery of or development of the green fluorescent protein from jellyfish, which I guess he got a Nobel Prize.
Got a Nobel Prize.
Yeah.
Yep.
So I think this is great that there's going to be a regular effort to promote fundamental, just discovery-driven, curiosity-driven science in a venue, well, a political venue. Say, look, guys, wake up.
Yeah.
Mm-hmm.
Cien admits cynicism, suspects that the award is just preaching to the choir. I think
it'll get beyond the choir.
But it's because it's being presented by a congressman. I think that may give it actually some legs.
Right. That's what.
Maybe. If he picks the right stuff, right?
Yeah.
I mean, it's an important problem. I mean, basic research is being pushed aside, and you have a nice letter that was Rich that you brought up a long time ago that came out about this issue, right?
Yeah.
That's the Zerhouni letter.
Yes, right. That was
2005.
Got that there?
Yeah.
That was that came up a long time ago because we were talking about Norton Zinder's obituary, and his last publication was a 2005 letter to Science, co-signed by a pretty amazing crew of people, bellyaching about the diversion of funds from basic research into biodefense.
Right.
Mm-hmm.
And it's a big problem. When people it's a big problem. People need to understand what fundamental science is and how it works. And the notion of trying to, in particular, politically direct science or scientists towards a certain task is, if applied, as the sole way to do science, is dangerous.
Right.
We need to have a significant, it's not as if there shouldn't be some directed research, but we need to have a significant fraction of the research funding just to let people follow their nose, follow their curiosity.
Yep. All right. So Martin Chalfie, of course, is professor at Columbia University, which is where I am, and his colleague is the author of my pick of the week, and another professor at Columbia is Stuart Firestein. He's a neuroscientist, and he has written a very nice and short book called Ignorance, How It Drives Science. You can read this in 2 hours. Everybody needs to get this. It is really, really good. It's basically about how in science we don't try to make facts. We try and know what we don't know. We do experiments to find out what we don't know. And he spends a lot of time expounding on this. He uses 3 case studies of 3 different scientists who do this. And it's really, really good. So a lot of it is discussing what is a fact and how do we get it and and what good is a fact, and how the best science doesn't lead to an accumulation of them, but simply highlighting what we don't know. Really, really, really good. Part of the book, he talks about the Golden Fleece Awards. And he says, on the surface, a lot of these research topics look ridiculous. So he studies olfaction in, I forgot what the animal he used. Salamanders. All right. He said if William Proxmire had seen the title of his grant, he would probably think it's a Golden Fleece awardee. Mm-hmm. But in fact, there are very good reasons to work with model systems, and he complains about how we're moving away from this now as we move away from basic research.
I just bought it.
Really?
Had it delivered to my Kindle.
You'll love it.
I was looking at that and thinking I probably need to get a Kindle.
Firestein in himself is an interesting character. He started out in the theater, and he didn't get into science until very late, and his story is really interesting. But this whole idea of science isn't driven by facts is really, really interesting. And he also talks about how, we have to communicate science to the public. Scientists are obligated to do this because we use public money. And he says most of the time the public just gets facts, but they don't understand. They're not told, what the facts are for and what they lead to. And so
it's a byproduct of the way we teach science at the secondary school level.
Yeah, and he says he when he first started teaching here, he was teaching a neuroscience course, and he assigned this 1,500-page book, and he said every lecture someone would raise their hand and ask, is this going to be on the exam? Yeah. And he realized he was teaching facts, and that's not the way to do it. So he actually teaches a course called Ignorance, and it is really popular here at Columbia. And he gets different scientists from different areas to come in and talk about what you don't know about their subject area. Not what's known, but what you don't know. And this has really made me feel really badly because I think I just teach facts in my course, and now I want to change it. I don't want to just teach them what we know, but what we don't know. Anyway, it's a great book. I'd love to get him on and chat with him. So buy it. It's a little guy, and as I said, you can read it really quickly.
Great.
And not just Rich and Kathy and Alan, but everybody.
Everybody.
I'm not trying to make him rich or anything, but I just think it's really good, and you'll understand better what science really is. What's this quote, Rich? Who put that in? You?
Rich did, I think.
Yeah.
It's a classic. I had heard it before, but it's, yeah.
Are you going to read it?
Sure. I ran across this the other day. I love it. I love Richard Feynman. He's just terrific. Science is like sex. Sometimes something useful comes out, but that's not the reason we're doing it.
That's true. There was a quote in the Firestein book. It was, I don't remember who said it, but someone said, models are always wrong, but sometimes they are useful.
Great.
Right?
Mm-hmm. Absolutely.
Mm-hmm. Yes.
So stuff like that. All right. So we have a listener pick of the week, and this is, of course, from Adam, who is nominating Lee Van Valen as a pick of the week by way of his memorial site at leevanvalen.com.
And when you go to that site, there are a series of quotes that come up, and I captured two of my favorites. One is, it would be good to keep an open mind, but not so open that anything can enter unimpeded.
Nice.
And then scientific proposals are inappropriate for much theoretical work. When one knows just what one will do, it is done.
Yeah.
That's how an NIH grant is supposed to be, right?
Yeah.
Right.
You have to know exactly what you're going to do and how it will turn out.
And by the time you do that, it's done.
Right.
It's just all wrong. Can you imagine having a colleague like Van Valen?
It must have been pretty wild, yeah.
It's pretty wild.
I mean, if he was accessible, you could just walk in and sit down and have lunch and talk. It'd be cool.
Just from what I'm reading about him, he seems like the kind of guy who would have been up to chat about just about anything.
Mm-hmm. Yeah, somewhere in some of the stuff I read, it said he had something like 30,000 books in his office.
Whoa.
All right, I'm going to start a podcast with him.
Okay, when you figure out how to do that, that'll be pretty amazing.
Yeah, well, that's the key, right? All right, TWiV 199. You can find it at twiv.tv and at iTunes if you like iTunes and use it. Subscribe and leave a comment or a rating. It helps us to stay visible so more people will find us. We also have a Facebook page, facebook.com/thisweekinvirology. Send your questions and comments to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan, which I understand is in the Heartland. Thank you.
I guess you could put it that way.
Thank you, Kathy.
Well, thanks for having me. It's fun.
Rich Condit is not in the Heartland. He is at the University of Florida, Gainesville. Thank you, Rich.
You're quite welcome. It's great fun.
And Alan Dove is certainly not in the Heartland. He's in Western Massachusetts, where he tweets on Twitter and blogs at alandove.com. Thank you, Alan.
Always a pleasure.
And I am Vincent Racaniello, and also not in the Heartland. We're in the Northeast. And you can find me at virology.ws. And I want to thank everyone who wrote in on the website or on Twitter or on Facebook saying they do listen to the end. One of them said, hey, Kathy Spindler is not the only person who listens to the end. I do too. So for you, that's what this is. Thank you, because you're the only ones still listening. One guy said, I always listen to the end to see if there's an Easter egg. We have to give him an Easter egg at some point. We haven't had one in a long time. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Are you still there, Alan?
I'm still here.
You're low. Why are you low?
Why am I low? You want me to be high?
Let's see.
I'm high.
Yeah, dude. You want me to be high?
Dude.
Alan?
Yes?
Alan?
Yes, here I am. Here I am, giving you a level. Here is my level.
Good. Try that again. What's your name, Alan?
My name, Alan, is Alan.
Okay, good. That's good.
Alan, load of crap dove.
Alan, load of crap dove. Yes.
Okay, let's do it.